Alright everybody, so now what I'd like to talk to you all about are ketone bodies and the metabolism of ketone bodies. Ketone bodies are water soluble and they are a means for the liver to send energy-rich compounds to peripheral tissues, particularly when sugar levels are low. Now what they will do is you need oxaloacetate in order to oxidize acetyl-CoA via the TCA cycle. In the event that your oxaloacetate concentrations are low, the instance of like a low carb diet or something, your oxaloacetate levels are going to be low, in which case then you're going to be able to convert that acetyl-CoA into ketone bodies. These ketone bodies can be used by heart, muscle, and the brain. Your brain normally is going to use glucose, however, ketone bodies can be used kind of like a little bit of a last resort. Okay, now your most significant ketone bodies are these three. I'm starting at the bottom with beta hydroxybutyrate, acetoacetate, and acetone. Now what's significant about this right here is that acetoacetate and acetone, their relationship is that this is going to spontaneously happen, and that acetoacetate can be decarboxylated to form CO2, and acetone. Now both of these can be released simply by exhalation. Now, what we're going to primarily be focusing on, because we're going to be focusing on the synthesis and degradation of ketone bodies, is we're going to be focusing on acetoacetate and D-beta hydroxybutyrate. So ketone bodies, generally their concentration is fairly low. However, in starved conditions, they're going to be quite a bit higher. So in starved conditions or a low carb diet would be a good example, in which case, what your body is going to do is it's going to take TCA cycle intermediates and do gluconeogenesis and send that glucose to the brain. Likewise, it can take fatty acids, break it down to acetyl-CoA, and then that acetyl-CoA's availability can lead to an increase in ketone bodies. So basically, these ketone bodies can also make it to the brain. So think about it like low sugar or a low carb diet or starvation conditions. Well, then your body's going to say, well, we got to continue to fuel this brain thing because that seems to be fairly important. So one way is through gluconeogenesis by processing processing that acetyl-CoA and basically taking TCA cycle intermediates back up. Alternatively, you can go directly from fatty acids into increased ketone bodies or increased ketone body concentrations and send those to the brain. So that's under a low sugar condition. Okay. Now, in the case of someone afflicted with diabetes, well, that's insulin sensitive diabetes, where someone cannot basically someone cannot take up glucose efficiently. That's analogous to a starvation condition, in which case their ketone body concentration is going to go up, and as their ketone body concentration goes up, their acetone concentration is going to go up, and that's going to sometimes give off acetone has a little bit of a sweet smell to it. But one of the things that this can lead to, because if we talk about something like acetoacetate, that is acidic, and that's going to cause a depression or a decrease in your blood pH, in which case this could, if the ketone body concentration is extremely high, well then you could have something known as diabetic ketoacidosis, or DKA, which can ultimately be fatal. But in addition to that, as that ketone body concentration goes up, that can result in kind of behaviors like someone can become disoriented, frequent urination, that can ultimately lead to a coma and death. So one of the things that's a potential problem for is this can be mistaken for alcohol intoxication. And I remember a professor that I taught for in graduate school always talked about this, or he highlighted this as a potential risk because, if there is someone who has very high blood ketone levels or high ketone body concentration, well, they might seem like they're drunk, in which case, maybe they're to a point where they would get arrested or something like that, and then if they go spend the night in jail, thinking that they are just drunk and, sleeping it off or something, that could lead to something very fatal. Something very fatal that could ultimately be deadly. Okay, so first we're going to talk about ketone body biosynthesis, and then we're going to talk about ketone body degradation. The first step in ketone body synthesis is the reverse of the last step of beta-oxidation, because what we have are 2-acetyl-CoA molecules. We're going to use that lyase enzyme to remove one of the coenzyme A molecules. So coenzyme A is going to be removed from this guy right here. So that coenzyme A is removed by the lyase enzyme, and the 2 carbons that remain, these 2 that I've just circled, they are going to be transferred onto basically this carbon right here. So we have a longer chain. We have our aceto-acetyl-CoA. So this is nearly a ketone body. The only problem is that we've got our coenzyme A molecule on there. Okay, so we've got our aceto-acetyl-CoA. The next step in this process is to use the enzyme hydroxymethylgluteryl-CoA synthase, or HMG-CoA synthase. Now this HMG-CoA synthase is going to take our aceto-acetyl-CoA, and it's going to use another acetyl-CoA molecule. So another acetyl-CoA right here. And what we're going to do is take this carbon right here and form a bond with that carbon right there. So we'll be using water in order to do that, and we'll make this longer chain where we're taking a 4-carbon molecule and adding 2 more carbons to it to get us to this beta-hydroxy beta-methylgluteryl-CoA, or HMG-CoA. Now HMG-CoA is a significant molecule because of the fact that it plays a pretty major role in cholesterol biosynthesis, in addition to ketone body biosynthesis. Okay, so after we formed our HMG-CoA, in order to complete ketone body biosynthesis, what we need to do is take our 6-carbon molecule, our HMG-CoA, and actually break off an acetyl-CoA molecule. Okay, so we had a total of 3 acetyl-CoA molecules input. 2 of them combined initially. We added our 3rd one to make our HMG-CoA. Now what we need to do is get one of those acetyl-CoA molecules off of there. So the enzyme hydroxymethylgluteryl-CoA lyase, HMG-CoA lyase, is responsible for that. So it's going to break the bond between carbons number 2 and 3 in our chain, and that is ultimately going to release our acetoacetate. Now it's kind of funny because earlier we were looking at this as when we combined our 2 acetyl-CoA molecules, we made something that looked like acetoacetate, but the problem was that it had that coenzyme A molecule attached to it. So in order to remove that, it actually took quite a bit more. So when we had our acetyl-CoA, it wasn't just as simple as remove the CoA group, and there you go, you've got acetoacetate. Instead, what we had to do was make HMG-CoA in order to remove a complete acetyl-CoA molecule. So once we've done that, we've basically got one of our ketone bodies that we're looking for, and that's that acetoacetate. Now, what that all boils down to, and what that is kind of simplified as, is we have our first step, our first step in this process is the same as the reversal of the last step in beta oxidation. So using that thiolase, we make aceto-acetyl-CoA. That aceto-acetyl-CoA, you have this aceto-acetyl-CoA, then we use HMG-CoA synthase to make our HMG-CoA. That HMG-CoA is significant because, like I said earlier, it is involved in cholesterol biosynthesis. So think about that as an intermediate. And it can be used for other things. But whenever we make that HMG-CoA, we can go in the direction of cholesterol biosynthesis, or we can just complete the production of our acetyl-acetate. Now, once you have that acetoacetate, and you've also released that acetyl-CoA, well, you have formed one ketone body, but now what you can do is you can take that acetoacetate and the enzyme beta-hydroxybutyrate dehydrogenase, add in some NADH, and ultimately generate your beta-hydroxybutyrate. So these were the three ketone bodies that we talked about, and what those three ketone bodies were. So these were our three different, just a second. These are our three different significant ketone body molecules, and so primarily acetoacetate and beta-hydroxybutyrate are the ones that you need to be concerned with, because of the fact that those can be transferred to brain and skeletal muscle and be utilized to actually extract some energy from. This acetone is entirely possible, or entirely likely, that what's going to happen to it is it's simply going to be exhaled. Okay, so now that's all our breakdown. That was all our synthesis. Here's our breakdown. Now, imagine that beta-hydroxybutyrate actually makes it to the brain. What's going to happen to that? What energy are you going to get from it? Well, one thing that you're going to do is you're going to use the enzyme beta-hydroxybutyrate dehydrogenase. That single enzyme is going to help you produce some NADH. So let's think about NADH in terms of, well, that's going to go on to be used for, that NADH is then going to go into the electron transport chain. So ultimately that will feed into generating our proton gradient so that we can ultimately get some ATP out of it. Okay, so then that acetoacetate molecule that results, so sorry, beta-hydroxybutyrate, that being a primary, or an example of a ketone body, that's going to help you get some ATP, or that can be used as an ATP source because it's going to help generate NADH. Acetoacetate, that is not necessarily going to be very helpful right off the bat, but rather what you're going to have to do is metabolize it a little bit more. So you'll use the enzyme 3-ketoacyl transferase. Now what that is going to do is going to use a succinyl-CoA molecule and basically transfer our coenzyme A molecule onto acetoacetate to make acetoacetyl CoA. So this enzyme right here, this 3-ketoacyl CoA transferase, what we're seeing here is a conversion from succinyl CoA to succinate by way of transferring that coenzyme A molecule onto our acetoacetate. Okay, this might seem a little bit familiar, and the reason for that is because, or this conversion of succinyl CoA to succinate might seem a little bit familiar, and you'd be right, it is something that we've seen before, but what we've seen previously is this enzyme right here, succinyl CoA synthetase. We saw that in the TCA cycle. In some instances, you might see this referred to as succinyl CoA thiokinase, or succinate thiokinase. And this goes back to thinking about the TCA cycle and all those aniloporotic reactions where we were basically fueling other things. So by these two intermediates, the succinyl CoA and succinate, well, succinyl CoA serves as a metabolite involved in ketone body degradation. It's also an intermediate in the TCA cycle. So if you think about the pools of different metabolites in the TCA cycle, well, ketone body degradation is constantly taking away succinyl CoA and generating succinate. Okay, so that succinate needs to be replenished so that, well, not only can ketone body degradation continue to go on, but also so that the TCA cycle can continue. Okay, so essentially this is replenishes succinyl CoA. I'm just going to write it as S-CoA pools or concentration. So at any rate, acetoacetate is converted to acetoacetyl CoA. Now if you look at this, well, this is 4 carbons plus an S-CoA. Now the great thing about this is that this is basically the exact same, we talked about a 16 carbon molecule and breaking that down into, or 16 carbon fatty acid and breaking that down in beta oxidation, and if it's 16 carbons, we'd go through 7 cycles of beta oxidation. The last step would be thiolase. Hey buddy, come here. You want to say hi? Say, good luck on you guys' test. Otis. Okay. Sorry, this is my son Otis. He's been my helper. He's watching Puppy Dog Pals right now. So, Puppy Dog Pals. So any sort of abrupt pauses and me looking away, they're a result of me talking to him. So anyway, we have this 4 carbon molecule with our coenzyme A molecule attached to it. If we had 16 carbons and we go through 7 cycles, on that last cycle of fatty acid or beta oxidation, what we're left with is 4 carbons and acetyl CoA. Well, that molecule is acetoacetyl CoA. So that acetoacetyl CoA, can you say acetyl CoA?
What's that?
Well, that acetoacetyl CoA is this right here. It's 4 carbons in this coenzyme A molecule. What we need to do is use this enzyme thiolase to add coenzyme A to break this down into 2 identical acetyl CoA molecules. So we have these 2 acetyl CoA molecules, and these 2 acetyl CoA molecules, well, those can go into the TCA cycle, and the TCA cycle will allow us to get a bunch of FADH2, NADH, and everything like that. And we're allowed to do that, or not allowed, but we're capable of doing that because we were ensuring that all of our intermediates were consistently replenished. Alright, well that wraps up ketone body degradation and biosynthesis. Thank you all very much. Hope you have a good one.